Okay, good afternoon. So we're starting now, and today's class is about the other half of the hippocampus. So everything we talked about in the hippocampus and memory and patient HM and those deficits, those were real, but those are all about only half of the hippocampal kind of functions. And there's this whole other half of the hippocampus that we didn't talk about, so we'll talk about that part today. So here we have a picture of the rodent brain. And as you remember from the previous class, the hippocampus is this banana-shaped structure there. And note that it has this dorsal part more towards the top and this ventral part towards the bottom. And everything we said so far refers predominantly to the dorsal part. But there is this whole other half of the hippocampus, so what is that doing? Okay. So first, just to situate to how it looks like. So if you make coronal slice of the hippocampus that are oriented from anterior in the front to posterior in the back, you see this kind of structure appearing where the blue part is the hippocampus. So in the more posterior, near the back end, the hippocampus, if you cut like this, will look more like a elongated structure that goes all the way to the bottom of the brain. And we have been talking about predominantly about the dorsal hippocampus at the top, but not this ventral hippocampus at the bottom. So one first clue about what this ventral hippocampus could be doing came about from neural recordings of these cells. So we already talked about how recordings in the dorsal hippocampus showed that there were place cells which fired in particular locations in the environment. Okay, so if just to recap, it means that, let's say the rat is walking around this environment like this, there's some place cells are active in this particular location, and there's some other place cells that are activated in this other particular location of the environment, and then you can represent that graphically through this color map here. So in this example, like the first cell is a cell in the dorsal hippocampus that's active near the upper corner of the square environment. Okay, and then you see that that cell is active pretty tightly in that corner. So the higher firing rates, which are the pixels in red, are all concentrated together, and essentially the cell fires very little elsewhere. So a cell that has extremely high spatial resolution, like the maximum possible, would be something like all the spikes happen in one pixel of the environment and all the other pixels will be dark blue. So that would be the maximum amount of spatial kind of resolution a cell could have. And these cells are not like that, but generally speaking, most of the environment is dark blue, and the red pixels where the cell fires a lot are pretty few. So there are a few high firing pixels, and most of the pixels the cell is not active. And that's what the place fields look like in the dorsal hippocampus. But if you record in the ventral hippocampus, you see something very different. So first, see that, for example, this cell here, it doesn't have a lot of dark blue pixels, meaning there are very few pixels where the cell is not firing at all. But rather, the cell kind of is firing all over the environment and maybe has a preference of firing more in one location versus the other. So generally, what you see is that in the ventral hippocampus, fewer cells have spatial preference of firing, and the ones that do have very large place fields. So the red area is much larger in the ventral hippocampus than the dorsal, so they have kind of larger place fields. And the border of the place field is much fuzzier, right? So in the dorsal hippocampus, you can see that the cell goes from bright red to dark blue in a fairly small amount of space, meaning the cell is super active here and essentially not active when the rat is right there next to it. But in the ventral hippocampus, the cell is kind of more active here, slightly less active here, more active here again. So it's not as spatially tight as the dorsal hippocampus. So this suggests the ventral hippocampus might be doing something else. And another quick recap of the dorsal hippocampus was that when they made lesions of the dorsal hippocampus, it was observed that lesion in the dorsal hippocampus impaired contextual fear conditioning, so associating that this box is associated with the shock, so that association became worse, but auditory tone fear conditioning did not become worse. So the mouse could still learn that this tone predicts a shock as well as a mouse with a functioning hippocampus. So that's what happened if you lesion the dorsal hippocampus. And this suggests that the dorsal hippocampus is not generally involved with just producing freezing or fear or something like that. Because if that was the case, then the lesions would affect tone fear conditioning as well. So the fact that the lesion of the dorsal hippocampus affects only contextual conditioning suggests that that region is more related to the contextual representation. So rather than the emotional part of the contextual fear conditioning. So the thinking here is that the dorsal hippocampus is involved in understanding this context looks like this, and there's a table, and learning what this context is like. And without learning what this context is like, I can't really learn to context fear condition. So that's the reason why the dorsal hippocampus is involved in this, not due to involvement directly in fear or freezing or something of that sort. Okay, but the new data showed that if you lesion the ventral hippocampus, specifically this ventral CA3 region, you produce deficits in both tone and contextual fear conditioning. So notice that lesioning the dorsal hippocampus only affected the contextual fear conditioning, but not tone, as we saw before. But if you lesion the ventral hippocampus, you can get deficits in both tone and context fear conditioning. So that's something new, and it suggests that perhaps the ventral hippocampus is more generally involved with perhaps producing these aversive states or freezing or fear or something like that. Okay, so any questions about these data so far? Yep.
Did you mention on the previous slide that there was some sort of attachment to emotional valence when it comes to the tone?
What about previous slide?
I thought I heard you mention something about like emotional valence or some sort of relationship.
Yeah, so what I mentioned is that the dorsal hippocampus is likely not involved in emotional valence, producing freezing or producing fear, because the deficit seems to be only in contextual fear conditioning and not tone. Because if the region is involved in general production of fear, then the tone conditioning should be affected as well by the dorsal hippocampal lesioning. So this suggests the dorsal hippocampal lesion is impairing the learning of the context, not the emotional part or the producing freezing or producing fear. Okay, any other questions? But so far, yeah.
Would we say then the ventral hippocampus is responsible for the fear area, like feeling of fear?
Yeah, the ventral hippocampus, it does seem to be more involved with emotions in general than the dorsal. So, and that's what we'll get into in the next slides. Yes.
Thank you.
Okay, so that's one difference about the ventral hippocampus and the dorsal, which is that their place fields are bigger and fuzzier in the ventral. Also, ventral hippocampus seems to be involved in all tone conditioning and context, and the dorsal only in context. And also different genes seem to be expressed in the dorsal and ventral hippocampus. So here we have the mRNA expression for some different genes. And it's not important for us to know what these genes are doing, but just notice that there are some genes that are expressed only in dorsal or only in ventral hippocampus or both or neither and so forth. So these regions seem to be expressing a lot of different genes, the dorsal and ventral hippocampus, which also suggests they have some different function. Because generally when you see brain regions producing different genes, it's a good indication that they have some different function and may have some other distinct qualities as well. And another important difference is anatomically. So what is their connectivity? So here we have a summary of the connectivity of the dorsal and ventral hippocampus. And the dorsal hippocampus is mostly interconnected with regions like the medial and lateral entorhinal cortex, which are more related to spatial navigation and memory and things like that. And the ventral hippocampus is connected to the entorhinal cortex, but a different part of the entorhinal cortex, but it's also interconnected with a lot of regions that are known to be very important for motivated behaviors and emotions, such as the amygdala and the hypothalamus, and the bed nucleus of the stria terminalis is also part of the kind of extended amygdala, so it's also important in anxiety-type behaviors. And the nucleus accumbens is very important for motivated behaviors and pursuit of rewards and things like this. So just from the anatomy of the regions, you can also guess that these two subpoles of the hippocampus have different roles. And this is a very important way that anatomists have used to kind of suggest the function of brain regions even before the function is studied. So the idea is that let's say there's this brain region X that we don't know what it does. But you can see that it's interconnected with lots of regions that control like hunger and eating and things of that sort. So that suggests that region X is important in hunger based on the anatomical connectivity purely. And here, based on the anatomical connectivity, you'd think that the dorsal hippocampus is more involved in spatial navigation memory, while the ventral hippocampus is more involved with these emotional states, both positive and negative, right? Because some of these regions that's interconnected to are more involved with negative emotions like fear, and some of them with positive, rewarding emotions like the nucleus accumbens. Okay, so that's another difference between the dorsal and ventral hippocampus. And here we have another kind of slide showing the same thing, that they're interconnected to different brain regions. So overall it suggests that the dorsal hippocampus, again, is more involved with spatial learning and memory, which is what we talked about in the last 4 classes. And the ventral hippocampus might be more involved with the expression of anxiety and avoidance and the regulation of the hypothalamic pituitary adrenal axis. We don't need to remember the name, but this is basically the set of structures that controls the release of stress hormones in the blood. So maybe you have heard that, oh, when you're very stressed, then your cortisol levels go up and your heart rate goes up and that whole thing happens. So this HPA axis is this set of interconnected structures that controls this release of cortisol and similar hormones in the blood that cause you to become more stressed out. So the ventral hippocampus seems to be very involved in that. And its connection to the nucleus accumbens also suggests that it's involved not only in just negative behaviors like fear, but also in positively motivated behaviors such as pursuit of rewards. And we shall see both of those kinds of actions of the ventral hippocampus in this week. And before we get into how the ventral hippocampus actually controls these behaviors, we have to talk a little about how to name these different states correctly. Because in normal colloquial language, these words like anxiety, fear, and panic are used almost somewhat interchangeably. But in behavioral neuroscience, they have somewhat well-defined features that are different between those 3 words. Okay, so anxiety is typically used for describing the reactions to very low levels of threat. And that would be something like the first picture where nothing is really happening, but it's kind of a dark alley and it's at night and there's no one around. It looks suspicious, but nothing is happening. But there's some kind of potential threat that might appear. There's a higher probability of it being dangerous. And then in these situations that provoke anxiety, which is a reaction to the lowest levels of potential threat, we have behaviors like avoiding that location or eating later instead of eating now, or sleeping in a different location, or things of that sort. Okay. Then the threat level could escalate, and something like what you see in the middle picture could be happening. So imagine you're seeing the scene in the middle picture where there are all these people ganging up on some guys and beating them up, but they're not attacking you, but they're attacking some people over there, so it's still, unfriendly to say the least. And this situation is already a kind of more immediate sort of threat. There's something concretely there, and this produces this fear state. And in rodents, the most observed behavioral expression of fear is freezing, and they freeze to avoid the detection from a predator. So the predator can't see them because things that are running around and jumping are very visible, and things that are kind of not moving are harder for the assailant to detect. And lastly, you could have a super intense threat that is a life and death situation, such as if you're this gazelle thing, and the cheetah is eating you and biting you at this moment. This is the biggest threat you've ever seen in your life. So it's very scary, and then that produces this panic state, which can also be produced by some other life-threatening conditions like there's a fire or you're asphyxiating or things of that sort. So very high intensity, proximal, high probability threats. And that produces this panic state. And in the panic state, the most observed reaction, both in rodents and in people, is to escape from the panic by running away. So there are kind of three important things to notice here. So first, notice that when the threat is increasing, the behaviors that accompany that threat are changing, but they're not just changing as becoming more of the same behavior, but rather they produce three distinct different sets of behaviors, right? Because fear produces like freezing, but escape, which is in panic, is not more freezing. If anything, it's kind of the opposite behavior, which is more mobility because it's running. Or anxiety produces this avoidance of being in a place and eating differently, but freezing is not more avoidance. Freezing is like a different behavior. So these three different threat intensities elicit different categorical behaviors. So that's why they have these three different names. And this theory was developed here at UCLA in Professor Michael Fancelow's lab. So it's a very influential contribution to the field of fear. So any questions about how these states work? Okay, so these were developed predominantly to apply for rodents, but they occur in humans as well. So for example, when this freezing reaction happens in humans, it can also often be some kind of uncontrollable, kind of overwhelming behavior that sometimes doesn't even make sense and might even make you be in a more dangerous situation than you would otherwise. Okay, so historically, freezing made sense because you had to kind of not be visually detected by predators or people from the other tribe that are attacking you and things of that sort. But now there's a lot of occasions where freezing can produce a worse outcome. So for example, imagine like you're driving in the 405 at night and there's no one around, and then you see it's like a big truck coming in your direction, the opposite side, and is about to hit you, and sometimes people will freeze at the wheel, and then they get in an even worse accident because they froze at the wheel. So that's an example where now like freezing can lead to like a worse result rather than kind of minimizing the danger. Or also sometimes that can happen when people are swimming. So people that don't know how to swim, they flail about a lot in the water, but that's actually good when they're flailing about because then they don't drown. But if they freeze in the water, then they sink like a stone and they drown, and that's when it's dangerous. Okay, and from the point of view of the rodent, these behaviors make sense because those are kind of the appropriate reactions for being in the presence of a predator from different distances. So from the prey's point of view, the biggest problem that they encounter is like a predator. So let's say the predator is there, and I'm the prey here. So the first thing I want to do if the predator is far away is kind of to avoid that place. Like I'm thirsty, so I want to get the water bottle, but I'll just drink later, or I'll hide, or I'll stay near the corner. I'll just stay here near the corner so they don't see me as much. And then, if I go there, it's very noisy. If I stay here, it's like I stay in the corner so the predator doesn't see me. Now let's say I'm already here and the predator could see me. So then I'll try to freeze so that if I don't move, maybe it won't see me. But perhaps already is super near me, it's about to bite me. So if I freeze here and the predator is there, this is bad because then it'll eat me. So if I'm very near the threat, freezing is a bad reaction, so I should run away instead. And this kind of explains in a naturalistic way why these different behaviors are seen. Okay. So first, let's talk about this anxiety. Yeah.
When does fear produce the fight response?
That would be in this kind of last stage where the thing is already attacking you.
Panic?
Yeah. because the escape is the flight. And then the fighting would happen typically if the flight is not possible. So like if I'm here at a corner and the things that I can't escape, and that's when most often these rodents would be fighting. Yeah. Okay, so the first behavior they do is this anxiety, kind of avoiding the place, hiding kind of behavior. So here in LA, we're fortunate that there's not many mice around, but when I was in grad school in New York, in Manhattan, it's just filled with mice, just highways of mice crossing Broadway at night. And then you get to see what mice are doing in their kind of normal situation, which is they hide in these dark corners, like a mouse burrito or underneath like some gutter pipe. And that's a real robust behavior the mouse is doing. And we can kind of measure that in the lab. And the way to measure that is to put the mouse in a location that has some closed places and some open places. And the elevated plus maze is a very common way of doing that. So here we have a picture of this elevated plus maze. So there are these two open arms where the picture of the mouse is in, and these two enclosed arms. And in red, you can see the behavioral track of where the mouse walked around. And you can see it's typically more near the closed areas and not the open areas. And that makes sense from the point of view of the mouse, because if you let a mouse here and just release it here, you'll see that immediately, let's say I release the mouse here. The mouse will immediately go into a corner and stay near here where it's more safe from attacks from the open. And then they'll also go a little into the open to see what's around, because maybe there's some resources, food, maybe a lady mouse for me to talk to, who knows. So they also have some motivation to explore, because you can't just be hiding forever. But typically they'll spend more time near the closed areas than in the open areas. So this elevated plus maze allows the mouse to display both of those behaviors. And one thing when you look at a behavioral measure in a mouse that you should always ask for is, okay, the mouse does this thing of avoiding open spaces, which doesn't seem to be a thing humans do, because humans are not being preyed when they go into the light. But it's a thing the mouse does. And this is a way that the neuroscientists are studying mouse anxiety, which is really not what humans are doing when they're anxious. They don't avoid open spaces. So why do we think this has anything to do with the word that's used for human anxiety? Okay, so one way, and that's something you should always ask when you see any measurement in a mouse that's related to a human. It's like, why is this thing in the mouse related to the thing in the human that we're interested in? And there's many ways of validating that the behavior makes sense. So one way is pharmacological manipulations. Okay, so for example, there are some drugs that decrease anxiety in humans. So if you give a human a benzodiazepine, the human will be less anxious. Before that, the human will be like, oh no, I'll get fired from my job and there'll be so much traffic and everyone hates me. And then after that, maybe I won't be fired, who knows. And similarly, the same thing happens to the mouse. So the normal mouse, without any drugs, if they're in the closed space, they're happy, then they're going to the open space, like, this is open, something might eat me from above, I don't like this at all, it's very uncomfortable. And then if you give the mouse the benzodiazepine, the mouse goes more into the open space, like, maybe it's okay. I mean, my friend went to the open arm and he wasn't killed, so maybe it's okay, who knows. So you see that if you give drugs that increase or decrease anxiety in humans, the mouse will also, in the same manner, change its exploration of the open arm. So there's some pharmacological validity in the human and in the mouse in parallel in this behavior, suggesting something to do with the human. And there's also some other things that suggest this is something to do with the anxiety in humans. So another is that there's physiological parameters that go in the same direction. So the human that's more anxious will show an elevated heart rate and higher cortisol levels, and the mouse that's exposed to elevated plus maze will also show elevated heart rate, higher cortisol levels, dilated pupils, and reduced digestion, like all those things that the human with higher anxiety would also have. So there's kind of physiology that goes in the same direction, human and mouse. And there's also lifestyle kind of manipulations that go in the same direction in both species. So for example, a human that had a very nice upbringing and their parents were like, you are so wonderful, you can be anything you want, you could be president of the country. That person goes up to be super confident and just goes into the dark alley and is less likely to be pathologically anxious. And you have the human that's just beaten up by everyone and is getting a terrible life, all sorts of trauma. That human is much more likely to grow up to have higher anxiety and take fewer risks and hide more, etc. And that's also seen with these mice. So mice that grow up in kind of an enriched environment with lots of mouse friends and toys and different foods and activities and things to do, like those mice display less avoidance of the open arms than mice that are grown in like social isolation or maternal separation, or mice that had trauma in some way. So those mice avoid the open arms more. So we have all of these converging lines of evidence that suggest this behavior has something to do with anxiety in humans. And the reason I talk so much about why we should believe this behavior is because that's just something that we should generally think of whenever you see any behavioral measure in a mouse. Like, is that the thing that they're saying it is? So if so, it should have lots of parallels between what happens to a human with that kind of measurement and the mouse in the rodent equivalent measurement. Okay, so any questions about this part?
Regarding the car example you brought up, when you're like on the freeway and there's a truck coming at you, and some people freeze, there's also some people who experience both. They freeze
Experience
at
what?
first. There's some people that experience both.
Mhm.
They freeze at first and then make the proper movement.
Yes.
Is that inhibiting, so the three states are like keeping themselves in check to produce?
Yes, that's something we'll get into, but that is true. In some sense, because the behaviors that are seen in these three different states are not the same behavior, they also have to kind of mutually inhibit each other, because the mouse cannot be, for example, freezing and escaping at the same time. So those things have to be mutually inhibitory in some sense. And there are some circuits that have been found that do that, but yes, that is what's going on. Yeah, any other question? Okay. All right, so another way to measure anxiety in mice is with this high-tech box called the open field. So basically, if this is a box, and if you put the mouse inside the box, he'll avoid the center and go more into the corner of the box. So here's a track of a mouse walking around in this thing, and you see he spends more time in the periphery of the box. So periphery of box, closed arm, and center of the box, and open arms are measuring the same behavior. Okay, so now that we know what the plus maze does, let's see what happens if you lesion the ventral hippocampus. So first, on panel A, we have so in all of them, the closed arms are oriented vertically, and closed arms are the safe place. So the sham lesion mouse with a functioning hippocampus spends most of his time in the closed arms. Then if you lesion the dorsal hippocampus, which is the second panel there, the mouse continues spending more time in the closed arms. And if you lesion the ventral hippocampus, now the mouse loses part of its aversion towards the open arms, so it goes more into the open arms, suggesting it's less anxious than the control mouse. So this was one of the first pioneering papers that suggested the ventral hippocampus has something to do with creating more anxiety, because if you lesion it, there is less anxiety. So it's as if the mouse was treated with benzodiazepines, but it wasn't. It was just lesioned in the ventral hippocampus. And this is something that, I mean, it's not an experiment you can do in humans, but it's likely something that is affecting us as well. So perhaps there's this person who is less anxious than average, and one explanation might be that in that human, their ventral hippocampus is perhaps less excitable or less active than the human that has more anxiety. And we have seen in humans in kind of brain imaging tasks that there is activation of this ventral hippocampus equivalent of the human when the human is viewing like aversive, dangerous images. Okay, so another measurement is that if you lesion the ventral hippocampus, their kind of physiological reactions to open spaces also decrease. So the normal mouse with the functioning hippocampus, if you put that mouse in the open space, they will show an increase in corticosterone because they're very stressed out. And so corticosterone is the mouse analog of human cortisol. So they will show an increase in that hormone. And they will also show an increase in defecation when they're placed in these stressful situations because they kind of relax their sphincters. And then humans also do that. Like you might have, like some kid that goes in a roller coaster and then he pees himself on the roller coaster to the embarrassment eternally of all his friends. And that's because the human also relax their sphincters if they're super scared. Okay, so the mouse will show more corticosterone in the blood and more poop in the floor if it's stressed out. And then if you lesion the ventral hippocampus, both of those things decrease. Like if you lesion the ventral hippocampus and then put the mouse in the open space, they will show a smaller increase in both of these parameters. So also these physiological measurements of anxiety seem to be related to ventral hippocampus function. But that behavior that we're talking about in the previous week, which is the spatial navigation in the Morris water maze, that behavior doesn't seem to need the ventral hippocampus. So this was again this behavior where we had this bucket of water and then the rat is swimming around the bucket of water and then has to learn that there's a platform here for me to sit and avoid being swimming. Okay, so if you lesion the dorsal hippocampus, the mouse can't remember where the platform was, so it just swims around randomly and can't find the platform. But if you lesion the ventral hippocampus, the mouse is just fine. He finds the platform, so no deficit in this spatial navigation task. But it does have a deficit in this anxiety task. So we have this double dissociation where dorsal hippocampus is important for the spatial navigation, the water maze, but not for avoidance of open spaces. And ventral hippocampus is important for avoidance of open spaces, but not for this spatial navigation task. And it's also valuable that they did this kind of physiological characterization because you always want to see the manipulation producing converging evidence, right? Because from this result here, we see that lesion the ventral hippocampus produces more entries into the aversive, dangerous open arms. And one possible interpretation of that could be maybe the mouse is less anxious and that's why they go more into the open arms. This is a plausible interpretation. But there are many other reasons why the mouse could go more into the open arms that has nothing to do with anxiety. So it could be maybe lesioning the ventral hippocampus makes the mouse confused. So it doesn't understand this is the open arms. I mean, that's why it goes into open arms. Or maybe it's like blind, so it can't see which arm is which. Or maybe it's very hungry, so it's very hungry, it will go more into the dangerous locations. And you always want, ideally, the people that are doing the experiments to try to control for some potential confounds. So the fact that the same manipulation also decreases this completely different measurement related to anxiety, which is this corticosterone level and defecation, together with the open arm results suggests that the manipulation really was decreasing anxiety rather than making the mouse blind or confused or hungry or some other thing. Yeah.
Then can we conclude that the lesioning the ventral hippocampus is basically like lesioning the amygdala, in which they're not experiencing any fear or cannot experience fear?
No, it's not any. So, but the result is similar in the case that if you lesion the amygdala, they'll also go more into the open arm, so there's that similarity. But there are many regions that control any given behavior. So this behavior of going to the open arms is controlled by many regions, and those are two of them. But they don't have identical function in everything necessarily.
I see. Thank you.
Welcome. Yeah, so that's also important lesson generally from systems neuroscience, which is that all the behaviors are controlled by kind of a network of brain regions, not by this one region. And also the opposite is true. The vast majority of regions control more than one behavior. So then one of the earlier studies looking at the electrophysiological differences of the ventral dorsal hippocampus was done by me when I was in grad school. Okay, so, and the quick background here is that if you put an electrode in the hippocampus, dorsal or ventral, and you just record the kind of local field potential, just kind of like similar to the EEG, but inside a brain region, you see that the voltage fluctuation extracellularly is kind of going up and down like this, and there's this cyclical 8 hertz pattern. And that 8 to 12 hertz pattern is called the theta oscillation of the hippocampus, and that's seen in dorsal and ventral hippocampus. And if you look at it, it kind of looks like that. So it's this oscillation that goes up and down like this, and the cycle lasts like 1/8 of a second. So it's like 8 hertz. And the way to quantify kind of how much of each frequency there is an oscillation is done by this graph here called a power spectrum. So in this oscillation, the biggest thing you see is this 8 hertz oscillation. And that's why this 8 hertz peak is the biggest thing in the power spectrum. But there's also some power at some other frequencies, and that's because the oscillation is not a pure sine wave, but there's some other components in it. So for example, you see there's some fast oscillations there, right? So there's some fast things that are riding on top of this bigger 8 hertz oscillation, and that produces these peaks at like 30 or 15 hertz. And this kind of thing is the same exact power spectrum that you see sometimes in some audio players, that they show how much bass you have in the song, etc. It is doing the same mathematics but on music. So when you like boost the bass on the song, it's boosting these slower oscillations in the song. But this is what's happening to the hippocampus. You have this state oscillation, and you see that the spikes in the hippocampus are kind of locked to different phases of the oscillation. So you see this oscillation goes up and down and etc. And here we have the spikes of a cell in the hippocampus, and you see the spikes tend to happen at a particular phase of the theta cycle, showing that the spikes are being locked by this oscillation. And this oscillation is caused by synchronized synaptic input that's coming into the hippocampus. And this kind of thing is happening in all the brain regions. Like all the brain regions have some extracellular voltage fluctuation due to synchronized synaptic input that's happening. So when you go to like a sleep lab and you put like a cap on you that records your kind of electroencephalographic signals, like from the skin, those are these LFPs from brain regions that are being added over large signals until they come out in the skin. But so that's the version of this, but inside the brain, inside the hippocampus, looks like this. Okay, so that's what this theta oscillation is. And now, why do we care about this oscillation? So one reason is that it synchronizes firing within the hippocampus. So it gives the brain region sort of an internal clock to know, so that this cell knows when the other cells are more likely to be activated. Because, oh, we're all activated kind of in this part of the cycle, so let's use this common clock to coordinate our activity. So it allows the cells to more efficiently synchronize their action potential timing. And it might be used to propagate information to downstream targets. And the way this was shown through a correlative measure is shown here, where this is something done in the dorsal hippocampus, which is important for memory. So they made the mouse do this memory task where he has to remember which arm of the maze has the food. So when he's running, he has to remember, oh, I have to go to the left. Try to keep that in his memory. And then they saw that the synchrony of the oscillations in this theta range between prefrontal cortex and dorsal hippocampus increased when the mouse had this memory task associated with the movement. So this suggests that if two brain regions have a higher synchrony, it's an indication that two regions might be working together. Because if the neural activities of the two regions are going up and down together, it's an indication that perhaps those two regions are communicating with each other more, something like that. And that's another, the strongest candidate of what these oscillations could be doing in the hippocampus. It's to kind of communicate with other brain regions and synchronize firing within the hippocampus. Okay, so any questions about these oscillations or how to measure them or what they're doing? Right, so the ventral hippocampus projects to the middle prefrontal cortex with this unidirectional projection shown here. And both the ventral and dorsal hippocampus, both the ventral hippocampus and the prefrontal cortex are known to be important for this plus maze open arm space avoidance behavior, and they're connected, so perhaps these two regions are communicating with each other to guide anxiety behavior. So that's the question that we were studying. And to do that, we use this measure called synchrony of the kind of power correlation. So basically, if you have electrical signals from two regions, this black region and the blue region, if you have high power correlation, you would have, whenever the blue signal state oscillation is big, the black signal state oscillation is also big, so they are very positively correlated. This would be a high power correlation. And a low power correlation would be when the black signal state oscillation is big, the blue signal is not big, they're completely unrelated. So this would be a situation with low power correlation. So if there is higher power correlation, suggests there is higher synchrony between the two regions. So the two regions might be working together and communicating with each other. So that's the method that was used. And basically, if you record the signals from the ventral hippocampus and prefrontal cortex in a familiar, safe environment, shown in black, the signals are already pretty well correlated, suggesting that those regions are already talking to each other. But if you put them out in the open field, the correlation increases. Like those regions are now talking to each other more intensely, transmitting more information, etc. So you see this correlation increases from 0.5 to 0.67. And if you look at the correlation between the prefrontal cortex and dorsal hippocampus, you see that that correlation is always low and doesn't increase from familiar, safe environment to anxiety-provoking open field. So this showed that the synchrony in the theta range oscillation between the ventral hippocampus and the prefrontal cortex increases in the anxiety-provoking situation compared to the familiar, happy, nice situation, suggesting that perhaps those two regions are communicating with each other more strongly during anxiety, and that communication might have something to do with the behavior. But so far, this is all correlational, so there's no functional hard evidence yet, but the correlation does suggest has something to do with anxiety. Okay, so any questions about this part here? Yeah.
So no correlation. No, sorry. We have correlation, but we don't have. Like, we could say it's not exactly true. Right?
What's not exactly true?
That the two regions may be communicating with each other.
No, that they are communicating, you could say it's true. But if this communication has some role in the behavior, you can't tell from this.
I see. Thank you.
Yeah. Okay, so any other questions? Yeah. No, the power correlation doesn't involve measuring firing. It measures just the oscillation. So if the oscillation in one region is big simultaneously to the other region also being big, and they fluctuate their... It's like if the oscillation size co-varies across regions, then this power synchrony will be big. So it's like in this example here. In the first case, the sizes of the oscillations are correlated across time, the high power correlation. In this case, they are not. No, here they use a different measure, but that's too much getting to the technical weeds here. But it's a different measurement of synchrony across oscillations as well. Okay, so this shows that the ventral hippocampus and prefrontal cortex become more correlated with each other in the theta range during anxiety compared to baseline conditions. And also that we found that theta power, so the size of the oscillation also increases in the open field and elevated plus maze compared to the safe, happy, familiar environment. And that's shown by this power spectrum here. So you see there's now in the familiar environment, there's very little peak at 8 hertz, but in the two anxiety-provoking situations in red and blue, there is a noticeable peak at 8 to 12 hertz that appears, showing that the size of the state oscillation also increases in the anxiety-provoking situation compared to the familiar environment. And we also found that that size of the oscillation increase correlated with the animal's behavior in each of those mazes. Okay, so here we plotted how much the power increased in the y-axis, and the x-axis it is how anxious the mouse was. So here are kind of heat maps of the behavior. So this mouse here spent a lot of the time the center of the open field, so he wasn't scared of the center. And then this mouse here spent a lot of time the periphery, so this mouse was very scared of the center. And then what we see is that the mice that were more scared of the center, so they avoided the center more, they showed a bigger increase in theta power in the cortex than the mice that were not scared of the center. And similarly, mice that spend a lot of the times in the closed arms in the left, they also showed a higher increase in theta power compared to mice that went a lot into the open arms. So mice that show a higher amount of anxiety seem to have a higher increase in theta power, which suggests that this might have something to do with creating the anxiety. Yeah.
I was going to ask, what region does this theta power correlate to? Is it both the middle?
This is in the prefrontal cortex. Yeah. But it was in the hippocampus too, but this graph is in the prefrontal cortex. Okay. Then another thing we saw is that if you record cells from the prefrontal cortex, there were lots of cells that seemed to be correlated with the anxiety task components of the task. So here we have one example cell that fired almost exclusively in the closed arms. Then there's another cell that was active almost only in the open arms, and another cell that was activated only in the center of the maze. So it seems like these cells in the prefrontal cortex are active in either closed or open arms of the maze, which are the safe versus less safe parts of the environment. And then we saw that, there were some cells that were good at differentiating open versus closed spaces in the maze, such as this cell here in the top, fired more in the closed arms, and this one fired more in one of the open arms but not the other, suggesting it's not really differentiating open from closed arms. And these cells that were good at differentiating arm type in the elevated plus maze also happened to have action potentials that were locked to the hippocampal oscillation. So we recorded the oscillation in the hippocampus and the spikes of individual cells in the prefrontal cortex. So you see that the cells in the prefrontal cortex that were more like this, cells that differentiated well between regions in the plus maze, those cells tended to show more synchrony with the ventral hippocampal oscillation. So this upper gray cell was good at separating closed from open arms, and that gray cell, its spikes were very locked to the hippocampal oscillation. So the oscillation tended to happen at the peak of, the spikes tend to happen at the peak of the oscillation. And in this other case, we have this cell that doesn't separate well one arm type from the other. And that cell, its spikes were not very locked to the oscillation. So one spike occurred at the peak of the oscillation, another spike occurred at the trough of the oscillation. So this suggested that the cells that were representing open versus closed spaces in the plus maze might be doing that by receiving contextual input from the ventral hippocampus. And then they use that contextual information to differentiate the open areas from the closed areas. And so this was then causally shown by a follow-up paper where they used optogenetics. And there they showed that if you optogenetically inhibit the projection from ventral hippocampus to medial prefrontal cortex, you get a increase in open arm exploration. So this was going from the correlation to the causal. So just to reiterate what's happening here, because it's a little complicated. So basically, we have this elevated plus maze within which there are some closed areas and some open areas. Okay, so let's say I have the maze here, and these are the open arms, and these are the closed arms. So some of the cells are good at differentiating one arm type from the other. So perhaps one cell was active only here. So this cell was active only in the closed arm, so it was good at differentiating which one arm type from the other. And perhaps another cell was active somewhat homogeneously in the whole maze. Okay, so that cell wasn't good at separating one arm type from the other. And you also could have the opposite, like another cell could be good at separating arm type because it fired mostly in the open space and not the closed. But both orange cells are good at separating one arm type from the other, and the yellow one isn't. Okay, so then what we saw is, if we have the ventral hippocampus state oscillation like this, so this is the ventral hippocampus state oscillation, the cells that were good at separating arm type, which are these two orange cells, their spikes were very synchronized to this oscillation, meaning the spikes occurred, say, mostly near the trough of the oscillation. So it's like they're listening to hippocampus and I'll fire now. And this yellow cell was active in all sorts of random time points relative to the oscillation, and it seems to kind of not be paying attention to this oscillation. So that's the result that's shown here, which suggests that the cells that are representing contextual anxiety-related differences, they're using this hippocampal input to generate their internal representation of where is open arm, where is closed arm. Yeah.
For that explanation, are those neurons acting like place cells, or are they place cells?
It's a very good question. So are they acting as place cells? So it kind of depends on what you mean. So they have some placiness in the sense that they prefer being active in this place and not the other place. But they're not like place cells because they're not really representing that space exactly. They're kind of representing openness. And we did some controls to show that. Because you're absolutely right. From this data, it could be they're like place cells and not open cells or closed cells. And so this is a very insightful observation. And then to control for that, we did some manipulation. So one was, let's say this is the plus maze, and I'll just decrease the volume because it's generating some problems there. Okay. So the top arms were the open, like this. Okay, so let's say one cell was active here in this open space. Then if you put the same mouse and record from the same cell in the open field, which is different environment, different box, different day, everything, open field is here, this cell was now active here, which is the open space of this other different environment.
So is it better to say that they're more like contextual, like affected by the context and not the place, like place?
Yeah, so they are kind of representing openness, not exactly place. But the openness correlates with place. But you know it's openness because if you put it in a different space, it's not the same space, then they maintain their firing properties, kind of. Yeah.
So, are we saying that neurons that fire specifically in like the open versus closed arms, the open arms are the ones that have high theta correlation?
The open and the closed.
Okay, so why would it?
But not the ones that fire sort of uniformly everywhere, like this yellow cell. Or another possibility is it could fire in one open arm like this. But this cell that fires in one open arm is also not representing openness. Because if it was, it would fire here. So cells that fire uniformly or in one open arm or one open arm and one closed arm, something like this, they're not representing openness or closeness. So these cells were not synchronized with the theta oscillation. And these, regardless of them preferring open or closed, but they have to prefer something, those cells were, yes, more locked to the theta oscillation. Yes?
So why does it make sense that the closed arms would activate in the PFC and have high theta correlation with the ventral hippocampus? Because we're trying to talk about anxiety, right?
Mm-hmm.
And it makes sense that the open arms would induce anxiety in these mice.
Yes.
So why would it make sense that those would also have a really high correlation with the anxiety pathway if it's the closed arm? Shouldn't the mice not be anxious then?
Yes, but these are not representing more anxiety. They're representing the parts of space that are safer or less safe, and then you need to have the differentiation to know where I should be anxious.
So it's just a differentiation then, like.
Yes, they differentiate safe and aversive areas.
Thank you.
Yeah. Yeah.
So for like the cells that prefer open versus closed arm, do they show the same type of phase locking, or does the phase locking differ?
Yeah, they weren't more phase locked than the closed.
And like, what about the areas like of the troughs?
What do you mean, what area?
Like on the.
Oh, if it's in the trough or.
Yeah.
No, we didn't find any difference in that either. But we didn't look at that super carefully, so perhaps there's some subtle difference. But they weren't overall more synchronized in the closed or open. But overall, these two oranges are more synchronized than those two yellows. Okay, so any other questions about this? And this was kind of very exciting at that time because it kind of suggested what sort of information was going from one region to the other to construct a internal representation of where is safe versus where is less safe. But it was kind of 2010, so there weren't any causal manipulation tools to see if that projection was indeed important for the behavior. And then some years later, a follow-up paper did that where they optogenetically inhibited the ventral hippocampal projection to medial prefrontal cortex and saw that indeed activity in that pathway was important for the behavior, because if you inhibit that projection, the time in the open arms increases. So it's as if the mouse becomes somewhat less scared of the open arms and then goes there more, and also goes more into the center of the open field. So this is nice because we have these converging evidence again, where we have two different kind of boxes with different types of open spaces, and then inhibiting that projection seems to have the same effect in both of these behavioral assays. And they also show that the kind of latency to eat food in a novel environment also decreases. And this is also a different measure of anxiety. So this is, you put the mouse in this open field type box, and then he doesn't want to go into the center because it's bright lights and open spaces and who knows what will happen. And then they put some food in the center. And then the mouse is here, and the more anxious mouse will take longer to go to the center to eat the food. That's the latency to eat. And they found that this latency to eat decreases if you inhibit this projection. So we have kind of 3 different behavioral measures showing that activity in this projection is necessary for creating the baseline level of anxiety. Yeah.
What is the percent center distance mean? Does this mean that the green one was further away from the center or closer to the center?
Oh, that means the percent of the behavioral track that is, so that distance that's in the center. So it's like a mouse that explores like this and goes one time in the center. It has a low percent distance of the track in the center. Okay, so, yeah.
Just a clarification question regarding your study on the last slide.
Mhm.
So basically, all you're trying to say is that the signals, the strong signals we're getting at the closed spaces,
Mhm.
at the closed arm type,
Yeah.
it correlates to the troughs, the theta troughs.
Oh, you mean this one? This
Yes,
slide?
Yes.
Okay.
So the closed space,
Mm-hmm.
it correlates to the ventral hippocampus theta troughs.
No, not exactly.
Accurate.
No. So this theta oscillation is happening all the time, let's say in the ventral hippocampus. But then what we see is that some cells like that one might be more active in the closed arm. Or some other cells might be more active in the open arm. So both those cells that are separating arm types, they have some preference. Both of those cells have spikes that are more synchronized to the ventral hippocampal input, regardless of where they're firing in the open or closed arms. Because this oscillation is happening all the time, in the open or closed or center.
Oh, regardless. Okay.
Yeah,
Thank you very much.
welcome. Yeah, so any other questions? But yeah.
These cells, they're medial prefrontal cortex cells, right?
Mm-hmm. Yeah, so it recorded the oscillation here from the ventral hippocampus and the cells from middle prefrontal cortex. Yeah. And again, it's not something you can do in a human, but in a human we know that the middle prefrontal cortex is a region that has been implicated in anxiety disorders and that it is activated in humans when they're in the scanner going through some anxiety-provoking situation. Yeah.
So this study talks about the MPFC cells, but the study afterwards is talking about the cells from the ventral hippocampus to the MPFC. Is it the same cells we're talking about? or-
Approximately. So both papers are about the projection from ventral hippocampus to middle prefrontal cortex. But in this one, there's no manipulation of the projection. It's just recording the electrical activity from both regions and trying to see if they are correlated in some way that makes sense with the anxiety. And so this is all correlational evidence. Yeah.
So when you say more synchronized to the
Mhm.
ventral hippocampus, that means like more theta power.
Yeah, there's two different things that happen. So one is that, which is this, which is that the sizes of the theta oscillations on both regions become more correlated in the elevated plasma compared to the happy place, which is shown here. So it's like in the safe environment, they have a lower power correlation, and the anxiety-provoking environment, a bigger power correlation. So the sizes of the ups and of the theta oscillations become more correlated in the anxiety situation. That's one synchrony, and the other synchrony was here, which is that individual, and that first result has no individual cells. It's just oscillations in both locations. And then the second result, which is the more interesting one, is that individual cells in the MPFC that were better at contextually separating the safe versus aversive part of the environment, those cells were more phase locked to the hippocampal oscillation. So there's two different kinds of synchronies. Yeah, okay, so any other question?
I don't want to ask any questions, but
No, ask too many questions. That's the only purpose of having this class.
so if the cells are firing to the hippocampal,
Is that what?
if the cells are firing in synchrony with the hippocampal theta, and hippocampal theta is in synchrony with the PFC theta, why wouldn't we just say, oh, it's firing in synchrony with the theta and the PFC where the cells are in the first place?
Yeah, good question. It likely is. But the thinking here was that the theta oscillation in the prefrontal cortex is kind of it's being inherited from the ventral hippocampal input. Okay, so one advantage of this projection is that it's unidirectional. So the ventral hippocampus projects the prefrontal cortex, the prefrontal cortex does not project the ventral hippocampus. So then the expectation is that the prefrontal cortex would be listening, not the ventral hippocampus, because this is a very rare case where the projection is just in one direction. So the axons from hippocampus go to prefrontal cortex. So the more straightforward interpretation is that the prefrontal cortex is doing the listening and the ventral hippocampus is saying the things. And then we kind of were able to find some evidence of that as well, that the activity in one region was lagging behind the other region. Okay, so let's say you are copying me. So when I say something, you repeat it. So I say, I went to the park. Then you say, I went to the park. Now there's a lag between when I say the sentence and when you say the sentence. Because, the sound has to go there, to listen and say the thing. So the region that's following is expected to have some lag relative to the region that is doing the influencing. And then we were able to detect that lag much the same way we could detect this lag here. So let's say if you're repeating what I'm saying, so you are the output and I'm the input, then our biggest synchrony will not happen when we just correlate what you're saying with what I'm saying. Because you're saying what I'm saying like 2 seconds later. So the biggest correlations between our speeches will be my speech shifted 2 seconds into the future, or shifting your 2 seconds into the past. The biggest correlation will be produced when you were shifted into the past, not instantaneously, because there's a lag, because there's propagations. And then we saw that here, that the maximal correlation of the theta synchrony happened not with the instantaneous thetas of two locations, the two brain regions, but rather you had to shift the prefrontal cortex neural activity into the past a little to maximize the correlation, which suggested there's a lag. So that's why we think the prefrontal cortex was kind of listening to the hippocampus, not the other way around. Yeah, so you had a question?
Sort of. So are you saying that the area, like in the medial prefrontal cortex, it's not the theta oscillations there, but it's the theta oscillations that form as a result of the ventral hippocampus?
No, it is there, but it's there being influenced by the ventral hippocampus. It's like in this example, when he repeats what I'm saying, he is saying things. But that's being influenced a lot by what I am saying. So there is oscillation in both. So it's kind of like if you, let's say if the ventral hippocampal oscillation, let's say, looks like this, then we see that the prefrontal cortex oscillation looked like that. Maybe. I mean, this is an exaggeration, but you can see the orange one is leading the yellow one, like it happens before. So we saw something like this happening, but there is an oscillation in the prefrontal cortex, which is real.
But it's the same.
It's not the same, like this is an exaggeration, but it's like it's different. Like maybe this one has an extra peak here, but the maximum synchrony is observed when you shift the yellow into the past a little, because it's lagging behind the orange one.
Question.
Yeah.
So is it possible to measure like synchrony between different brain structures instantaneously, or do you always have to do it retrospectively?
Yeah, here we only talked about the instantaneous one, but it is possible to, and that's the default thing, is doing the instantaneous. But if you do this lag analysis, it gives this extra information of is someone listening to the other one consistently, and also by how much. And then that amount of lag should make some physiological sense, because if the lag you find is like 2 hours, then you did something wrong. So the lag has to be something, within some milliseconds, which is the known delay of synaptic pathways and etc. Yeah.
So in this example, the cells that we're talking about would be like in yellow, right? In the yellow area.
Yes.
And so would they be more synced with the orange or the yellow part?
They are instantaneously most synced to the yellow, but their maximum synchrony to the orange is shifting slightly to the past, exactly. Yeah, because both the local field potential and spikes in the prefrontal cortex are lagging behind their input. Okay, so any other question?
Can you plot the slides with the red and blue figures?
This?
Yeah. What is EPF?
What is who?
EPF.
Oh, elevated plus means.
What was that?
Elevated plus means.
Plus. Oh, okay. Got it.
Yeah. Okay. And then this follow-up study found that if you optogenetically inhibited the projection from ventral hippocampus to prefrontal cortex, the mouse became kind of less anxious in these various measures, showing that that projection really had some functional role in guiding the behavior, and it wasn't all correlation. Yes?
So here they didn't do it, but we would expect the synchrony to not be there anymore because we inhibited the pathway between them, right?
You'd expect the synchrony to decrease because you're inhibiting the input. I wouldn't say not be there, but less. Yes? Yeah.
So like the synchrony, overall synchrony plus this like specific synchrony to the specifically closed or specifically open, like the synchrony there if we inhibit the pathway decrease.
So could you say louder?
Oh yeah, so if we inhibit the MPFC ventral hippocampus pathway, then we expect to see less synchrony between the theta waves in the ventral hippocampus and the firing.
Yeah, you would expect that, yes.
Okay.
Yes. Yeah, because if you, like in this example where I'm saying the thing he's repeating, and then if you put a wall here to block the input, you'd expect him to listen less and repeat worse. Yeah. Okay. Then they kind of followed up on that finding we had that the cells were representing the open versus closed spaces, and tried to see what happens if you block the ventral hippocampal input optogenetically. So here they are recording from the prefrontal cortical cells in the elevated plasmase, and then, like in the first paper, they found, oh, some cells are active more in the closed arms, some other cells are active more in the open arms and represent the closeness, openness, and some other cells are firing randomly, not in ways that correlate with the anxiety parts of the task. Okay, and then to measure if a cell was good at separating open spaces or bad, they developed this measure called the EPM score. So basically, these two cells would have a high EPM score of maybe like 0.7, and this one could be like 0.8. And these cells that don't separate open arms versus closed arms well would have a lower EPM score, that perhaps 0.2 or 0.1, something like that. Okay, so this EPM score is something that's high for both closed and open preferring cells, and it's low for other cells. And those other cells, these yellow cells, were the ones that were poorly synchronized to ventral hippocampal input in the previous paper, and the orange cells were well synchronized to ventral hippocampal input in the previous paper. So the idea there was the ventral hippocampus is sending some information that's being used by those cells to generate this closed versus open separation, and that's why they're more synchronized to ventral hippocampus, because they're listening to that contextual input from the ventral hippocampus and then using that to assign this anxiety valence to it. So here they did this cool experiment where they recorded from the prefrontal cortical cells and recorded from the same cells in the elevated plus maze with or without ventral hippocampal input by optogenetically inhibiting the ventral hippocampal input. So for the first 5 minutes they recorded like in the baseline condition, the open in the plus maze, and the next 5 minutes they recorded from the same cells while blocking the ventral hippocampal input optogenetically. Then what they see is that the EPM scores during the light on period, which is when you're inhibiting the hippocampal input, those EPM scores are lower. So what this means is a cell that is like this, that fires more in one of the arm types, if you record from that same cell while optogenetically inhibiting the hippocampal input, this cell becomes more like this. It's like it loses part of its ability to differentiate open and closed spaces. So these orange cells become more yellow if you inhibit the ventral hippocampal input. So this was really cool because they showed not just that, this input has some role in guiding the behavior, but what is this input? Like, what is the MPFC doing with this input? So what it's doing with this input is generating these representations of where it's open and where it's closed. And then if you block that input, the cells are now worse at representing where it's open and where it's closed. And the mouse is also less anxious, suggesting that these representations of where it's open and where it's closed may be important for the mouse to distinguish where it should be scared versus not. So he's kind of less anxious when you inhibit that projection. So they have both this behavioral and the electrophysiological component converging on that model. Okay, so any questions about this part?
So the fact that like they have these like cells that can distinguish between open and closed, that itself is not necessarily related to like anxiety, but the idea is that like this may be used to like help the mouse understand where it is safe or not, so it kind of like helps with producing like anxiety.
Yeah, it's difficult to say the cells are representing anxiety because that's not a thing you can exactly directly measure, but the cells are definitely representing openness or closedness. And those are the parts of the maze that are creating the safety versus aversive feelings that in turn would generate this anxiety that you can't measure. Yeah, but that's the idea. Yeah. But also, one thing about this plus maze is that, so in your question, some of you said something like the open part is making it anxious, which is, but it's a little more nuanced than that, because it's not really obvious that the mouse is more scared while being in the open arms. Because you could interpret it in two ways. One way could be when the mouse is in the open arm, he's out here, then he's like very scared because it's the open, they could eat me. I don't like it. Okay. So that's one interpretation, he's more scared in the open. But it could also be the opposite interpretation, which is the one I favor, which is that this is not just exposure to threat, like the openness didn't jump at the mouse, but rather the mouse voluntarily chose to go into the open space at this moment, suggesting that that's when the mouse feels more confident, courageous, etc. So that it's like, I'll go and venture out there and see what happens. So my interpretation is actually the mouse is less anxious in the moment. He chooses to go into the dangerous part, right? Because if you look at the patients, for example, that have anxiety disorder, when they have more exacerbated symptoms, they stay more at home, get out less of bed, go less into places. They take fewer risks if they're more anxious. So my interpretation is actually that the mouse is scared more in the closed arm because it's choosing not to engage with the environment. Yeah.
I'm like getting really confused.
Mhm.
So this is not like the interpretation of the ventral hippocampus is not directly associated with the production of anxiety in the animals, right? Like
No, it is, but it's doing so, in this case at least, by providing contextual information input to this other region, and this other region is kind of assigning which region is safer versus not safe, and that's what might be creating the anxiety. But the dorsal hippocampal contextual input is not being used for this. So this region also represents contextual information like the dorsal hippocampus. But this contextual information is now being used for these different behaviors, like where it's safe or it isn't, and it's not being used for, well, where is the platform in the bucket of water? Like, it's not being used for that.
So specifically when you're talking about the projection from the ventral hippocampal to the MPFC, it's not like directly associated with the anxieties, but like other projections from the VSC to like other regions, for example the amygdala, that probably associates with the fear itself or the anxiety.
Yeah, that's a good question because we can't really say where the anxiety is being generated in the pathways, because it's more like a network phenomenon. It's likely not something like it's generated here, but clearly the ventral hippocampus is in that machinery that's responsible for creating avoidance of dangerous places, and the dorsal hippocampus is not. Yeah.
Is the medial prefrontal cortex like itself needed for anxiety?
Yes, it is. Yeah, that's the reason why I did this study, is because before we did it, there were 3 known pieces of information that if you lesion the prefrontal cortex, mice go more into the open arms. If you lesion the ventral hippocampus, they go more into the open arms. So both regions are functionally creating the same behavior. And also there is a projection from ventral hippocampus to prefrontal cortex. So the hypothesis was, well, these 2 regions seem to be doing similar things in this maze, and there's a projection between them. So perhaps this projection is how this phenomena is happening. They are using this projection to communicate with each other and generate the behavior of avoiding the open spaces. Yep.
Can we say that less EPM score corresponds to less phase locking? Can we say that? And then vice versa?
No.
Oh.
It does correlate with that, but one thing is not the other. But I think that's what you meant probably.
Yeah. Thank you.
Yeah, less EPM score is correlated with less phase lock.
But what it's trying to say is that we get a decrease on like arm preferences. They go all over the place rather than just stick with.
Yes, yeah. If you block the ventral hippocampal input, these 2 types of cells that prefer one type of arm, they become more yellow-like.
I see. Thank you very much.
Yeah, welcome. Okay, and since some of you mentioned the amygdala and the connection to that, the hippocampus, that's what was investigated in these Next set of studies. So if you remember from the slide about the anatomy of the two regions, the ventral hippocampus projects to MPFC, but it also receives a projection from the basolateral amygdala. The basolateral amygdala is the region that Professor Watson talked about a lot because it has some role in kind of reward-motivated behaviors, but it also has a much more deeply studied role in these aversive fear-type behaviors. And the basolateral amygdala projects to ventral hippocampus, and that's the projection that was studied here. So in the previous study, it was projection of ventral hippocampus to prefrontal cortex. And here is projection from amygdala to ventral hippocampus. So this is an input to the ventral hippocampus. And here they showed that if you optogenetically inhibit the projection from basolateral amygdala to ventral hippocampus, it also decreases anxiety. So the mouse goes less, it goes more into the open arms if you inhibit this projection. So both if you inhibit this projection to the ventral hippocampus or if you inhibit the projection from ventral hippocampus to prefrontal cortex, both of those seem to decrease avoidance of these open spaces. And that was seen both in the plus maze and in this open field. So again, good converging evidence in two different behavioral measures. And they also did the opposite result, which is if you excite that projection, then the mouse becomes more anxious and avoids the open arms more. So in the light off situation, they go somewhat in the open arms, and in the light on situation, they go less into the open arms. So inhibiting this projection creates less aversion of open arms, and then exciting the projection creates more aversion of open arms. So this is also very cool because you see exciting and inhibiting the same circuit create opposite behaviors, which is a very strong indication that they're really onto something. And that was shown both in the plus maze and the open field. Then here in this kind of control study, they tried to show that this effect was happening because the basolateral amygdala was releasing glutamate onto the ventral hippocampus. So the idea was we're optogenetically exciting the basolateral amygdala input to ventral hippocampus. So the basolateral amygdala releases some glutamate from the ventral hippocampus, and that glutamate that's released is causing this increasing anxiety. So to show that that was the case, they repeated that excitation in experiment, but at the same time, simultaneously, they infused the glutamate antagonists AP5 and NBQX in the ventral hippocampus. Okay, so the idea is you have the BLA. BLA has some cells that synapse onto the ventral hippocampus, and they release some glutamate in there. And if you optogenetically excite this, it releases more glutamate, and this glutamate action on the ventral hippocampus creates this more anxiety. Then to show if that was the case, they repeated this excitation experiment while adding glutamate receptor antagonists in the ventral hippocampus. So now this glutamatergic transmission is kind of impaired in the ventral hippocampus. And in that situation, if you excite this projection, you don't get this increase in anxiety. So they also showed that not only is the projection modulating anxiety, but it's modulating through this specific neurotransmitter. Okay, so any questions about this one here? Okay. And the same group also showed that that same projection from basolateral amygdala to ventral hippocampus was controlling not only aversion to open spaces, but also social interactions. This is a different thing we didn't talk about so far. We talked about spatial memory and anxiety. Now there's this different behavior, which is social interaction. So basically, if you get one mouse and put another mouse that's new in the box, and the mouse is very interested, like, oh, what do you see? How did you see this movie? Have I told you about the time that they climbed out of the cage? And then they talk to each other, and they're very excited. And so they have some social interaction. But if you inhibit the projection from BLA to ventral hippocampus, they socially interact more. So lower activity in this projection leads to more social interaction, which is a kind of new function of this circuit. Yeah.
Does this correspond to possible, like, alcohol-induced increasing social interactions, or no?
Yeah, it depends what you mean, but I think you probably think the same thing as me, which is that there is some inhibition of social interactions due to like aversion, anxiety, etc. So if you inhibit anxiety, perhaps they'll socially interact more. And that is certainly a possible interpretation here, because this circuit, if you inhibit it, does lead to less anxiety. So perhaps the less anxiety could lead to more social interaction. So that's definitely a fair interpretation here. And then they also showed the okay, so they showed the opposite, which is that if you excite this projection, then you have less social interaction, and then that agrees with your interpretation, because maybe they're super anxious, so they're like, I won't talk to the other mouse, he'll make fun of my whiskers, I'll stay here. So maybe they're more anxious, so they socially interact less. And they also did some studies to show that if you excite those terminals of the amygdala ventral hippocampus, that might release glutamate there, as they showed, and that glutamate release would expect to excite those cells in the ventral hippocampus. And one measure of recent neuronal excitation is this production of this gene called CFOS. So is anyone familiar with this CFOS neural activation idea? Yeah? Yeah, okay. So basically the way to think of this CFOS is that it's a marker of recent neuronal activity. So cells that fired a lot of action potentials recently will have a higher production of this gene CFOS than cells that were quiet and not firing action potentials. And like a human, and they don't have to be firing action potentials at this moment, it's just they were recently active. So one way of thinking about this in human terms is that a sweaty, panting person compared to a not sweaty person, you'd guess which one was recently active and running around. It's the sweaty, out-of-breath person. Even if the sweaty, out-of-breath person is not active at this moment, that is a human marker of recent human running around activity. Right, so similarly, cells that fired lots of action potentials very recently, they have a higher production of this CFOS gene. So their idea here was that if you excite this projection, releases glutamate, and the glutamate would excite these cells. So right after the optogenetic excitation, these cells in the ventral hippocampus should produce more of this CFOS gene. And that's what they found, indeed, that if you optogenetically excite those terminals, they release glutamate, and then the glutamate release causes a increase in the CFOS expression. So they showed kind of a molecular correlate of this excitation as well. Okay, so any questions about this here? Yeah?
This is the light blue, is the light on, right? The excitation.
Yes. Yeah, so it's basically the CHR2 in the light on condition has more CFOS. But okay, so that's what we had for this class, and then next class we'll talk about some other more recent studies about the role of the ventral hippocampus in various motivated behaviors. Welcome.